Good afternoon, my name is Matvii Tymchenko. I am from Kropivnitsky, Ukraine, and I am a pupil of Central Ukrainian Scientific Lyceum of the Kyrgyz Regional Council. My research supervisor is Elena Holodayeva.
The topic of my project is modeling and investigation of potential inhibitors of human aquaporin-4 under pathological brain conditions. The aim and objectives you can see on this slide. Disorders of the CNS caused by various factors remain a serious healthcare problem, leading to brain edema, BUB disruption, vascular dysfunction, and activation of aquaporins, particularly aquaporin-4. Excessive aquaporin-4 activity complicates treatment by hindering drug penetration into the brain, which results in abnormal water accumulation in neurons. Aquaporins are a family of membrane water channels that regulate water transport and are present in nearly all tissues and biological systems. It has been discovered that 3 aquaporins present in CNS, aquaporin 1, 4, and 9, but aquaporin 4 plays the key role in CNS pathogenesis. At the structural level, aquaporin 4 is unique aquaporin as it exists in 2 isoforms. That rainbow composed of 4 identical subunits. The central pore formed between these subunits is ampathetic. Such an arrangement ensures bidirectional water conductivity across the membrane. Aquaporin inhibition has proven its effectiveness. However, some studies of inhibitors have reported controversial results. A promising candidate is the natural flavonoid chrysin, which is capable of crossing the BBB and suppressing aquaporin-4 activity. Chrysin is also widely available in food sources. Flavonoid chrysin consists of 3-ring scaffold, stands out from other inhibitors with a wide range of beneficial effects, which you can see on this slide. The structuralographic structure of the aquaporin-4 was obtained from the protein data bank. Modeling modification was carried out using Chimera-X, and conversion into PDB-QT format was performed with Autodoc tools. Docking and interaction analysis were done with Autodoc, Autovin, and Python 3.7, while visualization was performed using Chimera-X, BVDS, and LeakBlood+. A total of 168 chrysin-based compounds were studied, which structures could be represented by the general formula. The evaluation of physicochemical parameters was carried out using SwissAdMe and Python via the RDGit algorithm. Toxicity risk prediction and drug likeness index estimated were performed using a series PROM. The modeled compounds were verified according to the compiled filter. Solubility was also estimated as an important criterion. Based on the boiled egg method, molecules that didn't match BBB penetration criteria fell within the yolk. Additionally, the Payne's filter was applied. As a result, from more than 160 possible modifications, only 53 were selected for the further study. At the next stage, selected models were analyzed in comparison with the parent molecule chrysin. It binded to the protein primarily with hydrophobic interactions. The impact of introducing halogen atoms, fluor, chlorine, and bromine at different positions was examined. Similarly, the effects of introducing a methyl, fluoromethyl, and ethyl groups were analyzed as illustrated on the slide. The influence of introducing several methyl, propyl, and their isomeric groups were investigated as well. As shown, almost all of the additional lipophilic chains generally led to decrease of the complex stability compared to chrysin. Paraposition was discovered at the best in complex binding. The introduction of the long chains into the ring has also had a negative effect on the complex stability. The introduction of 2 propyl chains with polar groups into ring B had a similar results. The addition of an extra ring in the meta and para position of ring B enhanced the stability of the complexes. The ligand interacted hydrophobically, completely blocking water entry. Introducing of the cyclopentalenes ring slightly weakened the protein-ligand complex. Hydrogen bonds were observed in less than half of all cases. When hydrogen bonds were formed with cysteine or methionine, a significant increase in binding energy was observed compared to chrysin. When hydrogen bonds were formed with histidine, the binding energy was not reduced. To better understand the modifications, we analyzed the effect of physiochemical parameters on protein-ligand binding energy. No significant correlations for them were identified. To sum up, 10 structures with the strongest binding to aquaporin-4 were identified. These compounds are predicted to cross the BBB and exhibit minimal risks of toxic effects. Therefore, they represent potential candidates for further in silico research at the main directions for structural modification in development of drugs aimed at suppressing cerebral edema. The full list of conclusions you can see on this slide. Thank you for your attention.